So, welcome to this lecture. This is a application oriented lecture, application of Raman spectroscopy in forensic science. In last class, I have talked about the application of Raman spectroscopy in medicinal field, and today we will be talking about the forensic science. So there is a word here, non-invasive detection. Non-invasive detection of concealed liquid explosive. Means you do not open the bag. So explosive, which is liquid, is hidden in some packet, and without opening the bag, you can identify the liquid whether that is of particular nature or not. So non-invasive means you don't have to do any kind of breaking or anything. You don't need to prepare the sample, and even if it is concealed, even if it is hidden, then also you can find out. So non-invasive detection of concealed liquid explosive. So now in this report, what they are studying is that H2O2. If there is a H2O2 hidden in this container, and you are not going to open it without opening, because you have seen in airport they don't open everything. They scan it, and they scan, and if they find it is suspicious, then only they open it. So without opening, if you want to know whether inside is H2O2 is there or not, then this is the report which suggests yes, you can find out. So now here they have develop some technique which is not a straightforward Raman spectroscopic technique that we use. Straightforward or conventional technique is that we excite, if it is the sample, we excite the sample and then we take the scattered radiation with the objective. So the scattered radiation is now taken back. So this is a surface modified technique. So in this technique what it does is that they excite in other place. They excite in the side, or from this corner, or from this corner. So they excite from any side, which means that you can excite from any side and you can take the spectrum from one particular direction, and then you can find out the Raman spectra of that species. So here in a bottle, if you have some substance, so which is somebody wants to know whether it is explosive, particularly in this study whether it is a H2O2, then this laser is exciting it, and there is a collection point, and then fiber optics cable, it is going to the instrument. So here what they found is that this jar only spectrum is this one. This is the jar spectrum, and this conventional Raman through the jar. Conventional Raman means this way. This is the normal technique. If you do, then you get this spectrum, and if you do this surface offset resonance surface offset Raman spectrum, if you take, then you get this one. What is interesting to find, the jar you cannot expect the band for the H2O2, but from conventional Raman, you don't find the band for the H2O2. What is the band for the H2O2? This is the expected band for the H2O2. And this band is absent in the Raman spectrum which is recorded conventionally. But this band is present in the Raman spectrum which is recorded based on the offset, surface offset. So this is the part is interesting. This is the jar spectrum. Obviously we don't expect any H2 to Jar means only this material which is making this jar. But conventional Raman is not giving any H2O2 band here. You see, there is no band here, but this surface offset, it is giving you the band. So this means you have a jar and you just focus on the jar, laser light, and take this. Use some mechanism where you will take that scattering radiation by collecting optics, and then you can tell whether inside the jar H2O2 is there and not without opening even. So this is very interesting observation, interesting finding. This one is another interesting one. Why it is interesting? Because this one, through this technique, you don't directly detect the explosive. So what you do, it is written detection of multiple nitroaromatic. You know, nitroaromatics there are some nitroaromatics which are explosive. TNT is also one of the nitroaromatic. So this is the 3-nitro group with a toluene. So this is TNT. So this is explosive. So this is under nitroaromatic. So there are multiple nitroaromatic explosives via the formation of a Janosky complex. Means you make a complex first, and that complex you make a layer on the surface, and then you record the surface enhanced resonance spectroscopy, Raman spectroscopy, SARS. So your main molecule to detect is this one. Your main molecule to detect this one, and then you make it react with this kind of enolate, and then it gives you this molecule, this anion molecule, anion with a thiol, anion with a thiol, and then this molecule you use on for the SARS measurement. For the SARS measurement, you use this molecule. This molecule is very efficient to make a layer on the metal surface and to detect through the Raman spectroscopy. So what is the Raman spectra? So now, so there are 2 point graph here. One is that this black one with TNT, and this is without. And there is clear distinction can be seen. See this area is more or less same, this is more or less same, but this area is clearly distinct. This peak clearly says that you have TNT inside the TNT as a material. So this is one example for detecting the TNT with a very high accuracy. So this is another molecule. Earlier was the toluene, and this is the amine group. So this is the one we have detected. This is another molecule. This is another molecule. And for every case, if you look at this reference one, here there is no explosive, and these 3 are explosive. So in every case, if you compare with this one, then you see in this one that peak indicating the presence of explosive that is there. And it's very clear. So this SARS can detect the explosive. Only thing is that you have to take out the compound, and then you have to react with that reagent, and then you make a layer of the SARS material and detect it very nicely. This one, last 2 slides were for the explosive, and this is for the drug. Forensic means explosive, drug, blood, all these things comes into the forensic area. So detection of drug, And how to detect the drug if from the fingerprint. Like if you have taken some drug and then you have used some door handrail or somewhere, then you have a fingerprint. From that fingerprint, taking the information whether there is a drug or not. So the main problem with this that with fingerprint, you have oil, you have this dry cell, as well as you have some proteins. From there, you have to find out the signature of the drug molecule. So that this can be done in a very efficient way using the Raman spectroscopy. So Raman says the Raman spectra obtained from the latent fingerprints. This is the A is the oily area, this one, and this is the solid particle. Solid particle is mainly the dry cell, the skin, dry skin. So there are some points which says that it is better. Now this one, in this they want to highlight some peaks like CH stretching and deformation mode, which appears in 2899, 2800. Oh, it doesn't have. Okay. So this one I was talking about 2000. So these are the peaks. You have to know that which comes from the oily sample, and solid particle pattern. This one is the flake of the skins that I have told you. So now this spectrum, this A and B, this says the crystals in this is a cocaine. This is I think somewhere is written. Is a cocaine analysis. This is a cocaine analysis. This one is the cocaine analysis. So you see this Raman spectrum obtained from the crystal of latent cocaine hydrochloride. So cocaine hydrochloride, if somebody has taken cocaine and if that cocaine is sticking to your finger and you have used that finger for any purpose, anywhere you are touched, then from that fingerprint one can analyze the cocaine. So cocaine hydrochloride crystals after 15 minutes of photobleaching. Photobleaching is mainly necessary for the bleaching of the fluorescence, and this one is that with fingerprint. And this is another one, nitroazepam. This is also a doping agent, also a drug. This is another drug molecule. So two drug molecules they have found that they can find out from your fingerprint. So quality of Raman spectrum, how to increase the Raman spectrum quality, because there is significant fluorescence background. If you see that significant fluorescence background will be there, some other also I will show you that significant fluorescence background, and how to make sure that from the fluorescence background you get the spectrum. Spectrum always comes from the Raman experiment, but if the sample is fluorescent, then the spectrum is hidden in the fluorescence. So one has to correct it. So for correction also, there are many people are doing just correction of the sample, correction of the background. Last I think 2 years back I went to a Raman conference. There was a section. There was a section in which three groups presented, and all three groups presented on how to analyze the Raman spectrum, how to get corrected Raman spectrum from uncorrected raw data. See, whole group in a very, I think one was from Netherlands, and I think group was quite big, maybe 15 people are there in the last slide they have shown. So whole group is working on how to get information from apparently non-informative spectrum. In many cases you will see that spectrum change is so small that apparently you feel like there is no change. So now those groups, they devote finding some numerical method, how to get data from that. There are many numerical methods, statistical method, because this kind of forensic or medicinal one, you cannot just like that publish telling that this is a cancerous cell just because 1 or 2 studies you are filling. No, you have to make a whole data set. Forensic also, you are trying to accuse somebody, you are trying to convict somebody, so you should have data set. So from almost nothing complicated data, you are telling something very important. So this analysis part is very strong. Many spectrum today also will see that it will appear like there is no change. From no change, yesterday also, you have seen, no, yesterday I just noticed that one when Professor Hiramasu was recording the glucose solution, he felt like it is a water. And then he initially thought it is water because strong water band and this OH and then bending. And then when somebody told, no, this is the glucose solution, then immediately he became careful because it appears like a water solution. But the person who has given the sample, he is telling, no, there is a glucose in it. And finally you see he got some glucose peak. Glucose, sucrose, jo bhi hai, that peak he got. So apparently the spectrum doesn't look like different. So analysis is very important. Only thing is that we don't care analysis. We just want to have very clear about spectrum and then only we speak something. But analysis is very important aspect in Raman spectroscopy. So this one is that this is again by SARS. In this case they have used an antibody. That antibody will bind with the protein and that one will be sprayed on the surface and then you do the SARS mapping. And they have found this antibody used SARS technique is very informative and it's detect very minimum quantity. Minute protein quantity also can be detected with this one. So this is the one they have coined antibody. This is antibody silver nanoparticle Raman probe conjugate. So ASNRP, yes, RPC, antibody silver nanoparticle Raman probe conjugate. So this is the technique they have used and they have found that this technique is allowing them to find very minute quantity of protein. So for that they have used one antibody immunoglobulin G, which is very well known. For many people study this immunoglobulin G and they have found a really interesting result. Now one thing I want to say that you see this is a kind of forensic science or biological science discussion is going on, antibody and all these things. But you see that report, whatever, this is from some report. So they have done so many, they have taken help of so many techniques. You see this is a electron microscope, this is UV visible spectroscopy, this is again, I think this is a scanning electron microscope, this is tunneling electron microscope, scanning electron microscope, UV visible spectrum, XPS, photoelectron microscopy. Now how is it possible? If it is done by biologist, because you have this protein and all these things, we cannot handle protein and all this antibody, protein. So if it is done by biologists, how come they do this one? This is in the domain of physicist or chemist, physical chemist. And this is a purely spectroscopy, XPS, photoelectron spectroscopy. It talks about that these electrons in 2p, whether it is a 2p electron or 3s electron, that kind of talking. Biologists have no idea. So this underlying importance is that if you want to really find out some good information, you have to make collaboration. You have to use various kinds of data so that it is not, your contribution is not, you should not highlight that it is my paper, so only I should do. If you want to highlight the subject, then you have to take care of the whatever all possible instrumentation is there, which will give you more and more information through which we can understand the system. Understanding the system is more important. That is why this kind of image is there. Again spectroscopy is there. Again spectroscopy. Again image. So everything is there. Whenever you see image, you must see this one. This is the bar. This is the length. What does it mean? Since you people are students, so this thing will be more important for you. It is a electrons beam scattering image. So through that you can see even nanometer scale. Optical, if you want to see through microscope, then your limitation is the light you are using. So if you are using this red light to see, maybe 800, half of that 400 nanometer that you can see. But this electron microscope you can see even nanometer. So this size is important, this bar is important. Now this bar says that this much distance is 500 nanometer. And this, what will be the distance of, what will be the size of this? You can approximately find out something. So usually they give bigger one. Here also bar should be there. There is no bar. Bar should be there. Bar allow you to understand the size you are looking at. So this must be, if it is 500, then this must be around 50 or something, maybe 50 nanometer. So if it is maybe 10 times, maybe 50 nanometer. So your size of this nanocrystal, or if whatever it may be, that is 50 nanometer. So bar should be given. So similarly, this is 1 millimeter. 1 millimeter bar you are seeing. It's a very not zoomed one. This is again 500. So you see this is 500. This is also 500. So this is smaller. This is bigger. So what I wanted to tell you that through this technique, they are trying to find out the information on protein, because this protein you have to find out the technique, you have to know that protein, because this explosive, they will be in touch with the protein. So unless you deduct the protein contribution, you cannot find out the true explosives Raman spectrum. So that is the same one. Now whenever you say I have detected, then obviously somebody will be willing to know that what is the capability of your detection, the limit of detection, how small you can detect. Whether large amount of, only when you have a large amount of sample in drug in the fingerprint, then only you can detect, or small amount also you can detect. So for that, this limit of detection limit, they said the detection limit is as small as 10 to the power minus 7 milligram. 10 to the power minus 7 milligram. So it's a very, very small amount. And how they come to this conclusion? They have recorded Raman spectrum at different concentration. So different concentration, you see this is a blank one. This is a blank one without this, without the molecule, without the drug. And then this is strong peak at 10 to the minus 1 milligram. And this peak intensity, you see it's decreasing. Even at this concentration, we see a peak. So what is the concentration? It says that 10 to the power minus 7. And usually we plot a graph, and then you will say what is the detection limit. And in this case, they have used 3 wavelengths to detect. And this bar you might be knowing, this bar, what is this bar called? Yes, these are the error bar. So error bar is usually necessary, but we do not give, but error bar is very much necessary if you want to make your data convincing. Because if the error bar is large like this much, then that data point need to be repeated. The data point has not much confidence. So one will not give much confidence on this data point. You see this error bar, this means this data, this point can vary from 6,000 to almost 13,000. 6,000 to 13,000, this can vary. Okay. On the other hand, you see this error bar. This error bar is very small. This is also small. These are also small. So the green light error bar is green line error bar. That means green means when you are using this frequency to detect, that error bar is very high. So maybe that's why this frequency to detect is better, because this error bar is very small, and this also quite a lot better than this one. So this is one technique where the mainly sample preparation is different. What they do, that you put the sample inside this tube, inside the tube, and it is a long tube, and then you measure the Raman spectrum. Raman spectrum of you see this entrance aperture is at 7 millimeter. So Raman spectrum of this one. Only instrumentation is different, and this one is the spectrum. So this spectrum of pure cocaine hydrochloride, this is the pure one, and this is the one with only 5% cocaine hydrochloride in other substance. Because hardly anybody takes pure cocaine. It will be mixed. So you have to make sure that this mixed one, the spectrum can be identified based on the pure spectrum. And here they say that this mixed one spectrum is quite lot same as the pure one. So that was the cocaine, and this is the heroin. Heroin also, you see that spectrum of heroin, this is why it is like this. What is the reason for this? Huh? Yes, fluorescence background. But from there they will get the information. Now if the drug is there on the cloth, that also can be found. There are reports on that. Through clothing also they could find out the signature of the drug molecule. And this is again from the drug molecule, and the clothing is, they can detect even if it is sprayed on the cotton, silk, wool. So all this independently they have studied. Because this will be used for the real purpose. So if you tell that this lab can detect the drug in a crime scene, then police will come, and police will come with a sample, say that sample is a woolen sample. If you have not studied previously, if you do not have a database, then you cannot accept the sample. You cannot use a woolen sample and you have a just data set for the cotton one, because you are going to help police for convicting somebody. So this one is that a few more different variety of this one, just to tell this forensic science helps in various way. Raman helps in various way to the forensic science. This one is that from the blood strain, one can determine whether it is a female or male. It is not that easy, as we will see, that these are the average, this red one, average blood sample of a female, this red one, and the green one is the average blood sample of a female. And they are, just don't see this one, just see this one, and anybody will think there is no difference between these two. So just by seeing the spectrum, it is not possible. These two spectrum, red and green, is exactly same. But when you subtract, then you see some differences. This is the subtracted one. Subtract one from another. If this difference is consistent, then you can tell, yes, we can somehow tell that there is a male-female difference. But this difference is not sufficient enough to tell that this technique is good for telling you that whether it is a male or female. These are for crime scene. You have a blood sample or you have something looking like a blood. You do not know whether it is a human blood. If it is a human blood, you do not know it is a female or male. If it is not human blood, you do not know whether it is a animal blood or even whether it is from the vegetable or something. So many types of studies are there. It is whether it is a human blood. If it is human blood, then whether it is a male or female. If it is a human blood, then whether it is a peripheral blood, means blood which is circulating in the body, or whether it is some menstrual blood, or whether it is a animal blood, or whether it is synthetic blood. So you will see that so many kinds of studies are there, and for each and every studies they have done lots of statistical analysis, lots, because it will give you at the end of the study, you have to tell the government that your report is a convincing one. Based on your study, you can tell that this is this, and you can convey. That is a scientific evidence. And when scientific evidence, if you are presenting to the court, then obviously the court has to accept. But do the scientific community accept your evidence? This researcher will be very critical that this kind of things is not good enough for convicting somebody. So that's why these statistical things are done. So this is a bigger way, just same thing. So now you will see that so much. First you see where are the difference in the band. This region there is a difference, and this region means this region there is a difference. Then 1210, 1230, 1210, 1230. This is the region difference. 15, 16, 15, 18. So these are the region for the difference. And statistical analysis says that this male or female, this red one is probably female, male and female, their spots are almost in the same place. So it is not distinct. If it is that red is here and green is here, then you can tell it is a distinct one. So this is not distinct. So it is difficult to do separation based on this data. This is another thing, so whether this is mainly the America, whether Caucasian and African American, they can be, whether your Caucasian is an old generation, so they can be distinguished whether this race has come from that race or not. So that also they have said that it is possible. And these are I kept. This is the instrument name, and always you have to give this information. So they have used a Leica optical microscope. They have used 20x objective lens, 20x that you see in the 20x, 100x, 10x, and this is a automatic stage. The radiation they have used, excitation radiation, is 785 nanometer laser light. Power is 4 milliwatt. And 20 used, this is 10-second accumulation times were used, 10-second accumulation, and I think 20 samples, 20 times is recorded. So this race difference, this is also not that much, but if you see this difference, then the some of the places you see significant difference. So through this they can have an idea about the race. So this one is search from the dried blood sample. Whether dried blood sample can be used for the Raman spectrum measurement, that they have reported this one. And this one is whether the blood is from human or animal. So they have taken the Raman spectrum of all the different types blood. This one is the human, then cat, chicken, cow, dog, pig, rabbit and rat. Important thing is that they have made this table in which that group is created, and this group shows, based on this Raman spectroscopic data, that human blood should be clubbed here in this region, cat blood should be clubbed in this region, cow, dog, rabbit, rat and many things, and unknown. So it's very interesting things that based on Raman spectroscopy you can identify if the blood comes to this region, this most probable class, then you have to tell this blood is from human, and everything is quite separated. So this technique is very nice to separate the animal blood from the human blood. Now if you have a red-brown spot somewhere, some cloth or anywhere, if you see a red-brown spot, then you can think it is a blood, dried blood, or it may be something different like a tomato, ketchup and all these things. So this technique is also helpful to say whether it is a human blood or something else. Lipstick, so many things they have studied. So makeup, cherry wood stain, chestnut, fake blood, cough syrup, lipstick, ketchup. So many things they have studied, and they said that yes, Raman spectroscopy can help you to tell that whether it is a dried human blood or not. So if it is a vegetable strain, that also can be identified from the blood. So this is the distinguishing the menstrual and peripheral blood. I have told you this one. So they have studied this one in different substrate. So if the blood is there on the carpet or wood or polyester, so imagine so many studies are there for this purpose, then only one can use it for the real field analysis. So this is for the semen analysis. And if it is a heterogeneous, then also it can tell multiple donor. And this one you see this so much analysis part. So much analysis part is there. And they have overlapped this one, and in some cases it is not overlapping, some cases it is overlapping. If it is considerable overlapping like these cases, then it means that it is the blood, human blood, or in this case semen. Now this one is the blood in presence of contamination. If you have contamination, this is a pure blood, this is a contaminated blood, that also can be identified. Even the blood is contaminated, then also it can be identified. This one is mainly to show you that you see that so many recording, can you imagine? That means so much time they are spending, so many samples they are recording, imagine. So much sample, each line is one sample. So these conclusions are out of a lot of hard work that we are saying. Here also just to show that so many times they are doing, even though this report is from a very famous lab. This report is from a very famous lab, but still they are repeating the experiment. This one I wanted to show you. You see, this one is with the background. This is with a background, and Raman spectrum, a dye sample in vaginal fluid from different donors. Average, this is the raw. This is the raw data, and this is after baseline correction. See, so much change. And baseline correction is not that just you correct this much baseline. That has some mathematical significance. Whatever polynomial you are using, why you are using that polynomial is not just that you select like that and then deduct it. It should have meaning. That is why there are some groups who does this one only, and they based on their group, those kind of formulation will be there in your machine. In the machine program, the program allows you to do this kind of operation. So where from this program comes? The instrument manufacturer purchase that software from them or from algorithm. This is called algorithm. So they write a program. That program, if you run on this spectrum, then you get a clear, acceptable spectrum. So there are some groups who are doing. Now imagine those kind of groups don't do. Then you don't get a good spectrum. But we never think of contributing in that line. One should think contributing in fundamental even if it is not prominent. So another spectrum I think I have. This is also, you see the fluorescence background. What kind of fluorescence background is not at all good spectrum. But after many corrections, you will see that nice spectrum comes. Okay. So this one, I think this one is the one. So this is with background, without background. So you do some corrections and then you see this one. So background correction is necessary, as Professor Hiramasu also said. So with this, I am finishing it because you have to appear for this examination also. We do not have much time. So soon we will have an examination. Okay. So if you have some short question, you can ask now. Any question?
Sir,
Yeah.
Sir, I have a confusion that what is the fingerprint oil? Is it any solution or the production of our sebaceous gland?
Yes, production of our own body secretion.
It is also being tested by the spectrum.
Yes, because finally, if you have a fingerprint, that fingerprint will have your own oil, this hand oil, and then protein, and then scalp. So everything they are separately studied.
So one more question. There is keratin protein in our hair too. So that is also different in every individual.
I did not discuss about the hair part.
Yeah, but
definitely there should be study. Some specific study has to be there.
Thank you.
If there is no questions, then let us go for the examination. Okay.